Thank you so much for having me. I'm very excited to be here. As it was mentioned, my name is Patrick Allard. I'm a professor at the University of California in Los Angeles. And today I'd like to share with you one of the questions that drive the work in our lab, which is whether we're inheriting a legacy of the toxic burden from ancestors. So to frame this question and understand why it's even important to ask it, I'd like to go back to the beginning and start with the dogma of molecular biology, the fact that DNA makes RNA, and that in turn, this RNA makes protein. And this very elegant, important, but simplistic view of how genes in our DNA are expressed and ultimately how they function has really illuminated how we understand the contribution of genes for our traits, so what we look like, also our behavior, and ultimately our health. This central view of the genes in our DNA explains why we're ultimately unique in this world, why we don't look exactly like anyone else on the planet unless you have an identical twin, of course. It also explains, because we share this DNA with our relatives, why we do tend to look more like them rather than other people. So to go back to the question, what we inherit from ancestors, well, from our relatives and ancestors, we inherit DNA. But it turns out we inherit more than just sequence of DNA. We also inherit another layer of information, a very complex layer of information called the epigenome. And so what I'll tell you about today is describe a little bit this layer of information called the epigenome that sits above the genome, but also tell you how the environment can deeply play into the regulation of this epigenome, and through that lead to inheritance across generations of these perturbations of the epigenome. So what is the epigenome? One of the analogies commonly used to describe the epigenome is a series of bookmarks. So these bookmarks help a cell interpret the genetic code and about 20,000 genes in the genome, and put some bookmarks, to tell which gene should be read and therefore expressed, and which DNA sequences or genes should instead be silenced and therefore not read. At the molecular level, we understand this epigenome to be made of three different categories. The first one is called DNA methylation, and it's the type of epigenetic modification that has been studied the most, and therefore we perhaps understand the best. But there are other types as well. The second type that we're starting to understand more and more, but it's very complex, is histone modifications. Histones are the proteins around which DNA wraps itself. And the modifications of this histone is very important in determining whether the DNA can be open, and therefore genes can be expressed, or the DNA is instead closed, and therefore genes would be silenced or repressed. And then the third important category are small RNAs that can also interfere with gene expression. And not only do you have these three different categories, but they're not standing alone here. They actually interact with each other, which really brings a lot of elegance and complexity to this extra molecular layer of information. And then the other thing that I want to say about the epigenome right now is the fact that we've started to understand through decades of research that this epigenome is deeply responsive to the environment. Things like environmental chemicals, of course, but also as varied as temperature or stress can all alter this epigenome. So what we're going to try to understand today is not whether the epigenome can be influenced by the environment. I think we have enough evidence about this. But whether these modifications of the epigenome that are caused by environmental exposures can indeed be passed on from generation to generation. So in sum, is our health right now conditioned by what ancestors were exposed to? And by ancestors, I do mean our parents, grandparents, perhaps even great-grandparents and beyond. So to address this quite complex question, we need three things to be able to really tackle it. The first thing is a model organism that will make this question more approachable and tractable. And this model organism that we chose in the lab is called C. elegans. As you can see it slither through the screen, it is a worm, and it's a commonly used genetic model system. And through decades of research in its biology and genetics, we understand very well in which ways it is not a mouse or human, but also in which ways, because of its biology and the way it regulates things like the germline or the epigenome, in which ways it is indeed quite unlike mammals, so mouse and humans, among other species, and therefore can be used to really inform and understand our own biology. It also has a short generation time of about 4 days, which makes all these multi- and translational studies a lot easier from just a time perspective. The second thing that we need is a cell type of interest. And the cell type that we care a lot about in our lab are the germ cells. So germ cells are the cells that eventually give rise to what are called the gametes, so the sperm and the egg. And we care a lot about those cells because any molecular information that's passed on from generation to generation is ultimately passed on through those germ cells that are the only two connecting cells between generations that go between an organism and the embryo in the next generation. And the last thing that we need is an environmental exposure to test in this paradigm. And the environmental exposure that we started with was bisphenol A, or BPA for short. We chose it for several reasons. The first one, it's extremely prevalent in the environment. It's found in a plethora of products as diverse as plastic bottles, the lining inside of food cans or soda cans, thermal receipts as well. And because it's found in so many different products, I've only named a few here, it's also highly prevalent in human populations, over 90% of the population in the US, but also globally. So that was one of the reasons. The other reason is because thanks to pioneering work from people like Dr. Dana Dolinoy at the University of Michigan, we've known now for a while that bisphenol A can indeed strongly impact the epigenome of mammals, in particular DNA methylation in mice. But also there's mounting evidence that it affects the other types of epigenetic categories that I mentioned. So again, the question that we're going to ask is not whether BP affects the epigenome, we already know that, but whether those environmentally induced, BPA-induced epigenetic modifications can indeed be passed on from generation to generation. So I'm not going to show you the actual data today, but I'll highlight some of the findings that we've made not only in C. elegans, but also we validated some of those results using an in vitro cell model, mammalian cell model. And what we found in some was a dramatic impact over several generations of BPA on germ cell quality. So those germ cells tended to die over several generations, and the cells that survived, the germ cells that survived, gave rise to embryos that were less viable than the controls. So exposing the worms, in human terms, you would be exposing the great-grandparents to the worms that we studied, indeed profoundly affected those germ cells. We looked at them in terms of their gene expression profiles that looked dramatically different. And again, I've already mentioned the fact that the quality of these germ cells was strongly decreased. And again, at least the idea that, or the concept that BPA could strongly affect the transcriptome, so the collection of gene expression in the germline, was also validated in mouse work. But when we started to look more closely, we found in the worm that over several generations, there was also a profound impact on the epigenome, in particular on one of the three categories of epigenetic marks that I mentioned, in particular the one called histone modifications. And so we saw in the germ cells that histone modifications were indeed profoundly altered. And this is one correlation that we made, right? We have this great-grandparental exposure to BPA, and we see actually two correlations. We see a decrease in reproduction, and we see a profound remodeling of the epigenome. But with the tractability of a model system like C. elegans, we can go beyond these correlations that are common in the field of toxicoepigenomics and actually start to establish causation. So the way that we did this is use genetic tricks available in C. elegans, as well as using drugs, pharma drugs, that have been targeted towards the epigenome that are commonly used in mammalian species, and show that it's not just a question of correlation, that if we somehow, through these genetic or drug means, reverse the impact of BPA on the epigenome, we can then completely reverse the impact of BPA on reproduction across several generations, showing that indeed this impact on health caused by BPA over 3 to 4 generations is indeed caused by its primary impact on the epigenome. So to go back to our original question, is our health conditioned by what our ancestors were exposed to? I think a tentative and maybe not the most optimistic answer is that, yes, there's enough weight of evidence, not just in a worm model, but there's also ample evidence now in other animal species, like, so I mentioned the worm, but also Drosophila, mice, zebrafish. And there's now starting to be mounting evidence in humans as well. So it is, using a weight of evidence approach, it is clear that the environment can indeed have some effects that remain across several generations. And this might be a doom and gloom kind of conclusion to a talk, but I'd like to think that thanks to the technologies that are arising, such as the ability to modify the epigenome in a targeted way, or even using the drugs that we used in our screens or in our experiments, we have the ability to at least reverse some of the effects of our toxic legacies that are running across generations. So to conclude, I'd like to thank the people that have been instrumental over the years to get to those conclusions. I'd like, of course, to thank my funding agencies, and you can always ask questions or make comments using the QR code on this slide, and also find us on Twitter as well if you want to interact through this media. Thank you very much.